Built-in mic. Okay, how about that? Can you hear me now? Built-in mic. There we go. I think,
okay,
yeah.
how about that? Can you hear me now?
Perfect. Okay, great. Awesome. All right.
There we go.
Okay, yeah. How about? Okay. All right. Excellent. Excellent. Okay, just to double check, I muted my own YouTube feed, but I want to make sure I don't mute everybody else. So can y'all still hear me? Okay, perfect. Awesome. All right, so I'm going to go ahead and get started because it is just a little bit after 1 o'clock. And so I tend to enjoy a rigorous relationship with time. So here we go. My name is Lee Ferguson, and I am the AP Biology teacher at Allen High School in Allen, Texas. And I'm going to be doing the review about labs. Now, when I say labs, I'm not going to go through every single lab that you might have done during the school year. I am going to go through some data analysis items and some of the labs that people asked about. And I got the list of questions earlier this week, and I'm not going to have a chance to address all of them. However, what I am going to do is put together a Google Doc that has answers to all of those questions. So in case you asked a question and it didn't get addressed, it'll be in that document. And so, as it says here, I'm linking that document below. I'm also going to put a link to this presentation in the description of this video so that after the stream gets recorded and is uploaded and archived or whatever to YouTube, you'll have access to that as well. And so what I'm focusing on here is the labs that had the most questions asked about them. And so just kind of an FYI, the labs that your teachers do belong to one of the 4 big ideas that the biology course is based around. And some of the labs weren't asked about at all. And so please, if you have questions about those labs that I didn't address, if we have time, I'll go back and try to address those items. But otherwise, I'm sticking to the labs that kids asked about the most frequently. And so those are the housekeeping items for this afternoon. And then how you're going to use this review, or the best ways to use this review, to kind of brush up on your data analysis skills, because those are pretty important in terms of being able to answer questions and produce work on the free response portion of the exam. But also, they're just good skills to have. To review your data presentation skills, and that's also, good skills to have in terms of communicating the results of your investigations. And then we're going to review some experimental design skills, because you may be asked on a free response question to either, design an investigation or to evaluate an investigation that somebody else has designed. And so it's important that you have those design skills or kind of know what those skills are. And some of the slides I'm going to show you are going to kind of jog your memory about the lab work that you've done during the year. But please understand that this is not a substitute for actually doing the labs. And so, what you're going to see is, like I said, I'm focusing only on the labs that kids ask the most about. And so hopefully, that'll kind of, jog your memory about some of the stuff that you did in class. And if it doesn't, well, hopefully it's a great introduction. So let's go ahead and get started. And so one of the things that I think it's really important for you to understand is that not every teacher does the labs exactly as they're written in the College Board AP Bio Lab Manual. Teachers do have the freedom to select labs they feel align to those big ideas, and as long as they align to those big ideas and build in the concept of inquiry, that teachers will do whatever labs they feel are best at showing those things to their students. And so what you're going to see is stuff that may or may not be representative of what your teachers did with you. And so be aware of that if I talk about a lab that your teachers didn't do with you, they might have done something similar and the concepts might have been similar, but the lab itself might not have been exactly what I'm describing. And so please keep that in mind when I talk about some of these labs. And so let's talk about the first thing that I want to go over with you are some data analysis skills. And so one of the things that kids asked about the most often was statistical analysis. And you have to be able to interpret stats such as the mean, the standard deviation, and notice there's an asterisk there, and I'll explain that in just a second, and the standard error of the mean, and also the 95% confidence interval. The reason the asterisk is there for standard deviation is that you don't actually have to know how to calculate the standard deviation, but you have to know its significance, and so we'll talk about that here in just a second. So let's talk about the mean. The mean, remember, is the average of a set of data points, and we use the mean as the starting point for things like the standard deviation, the standard error of the mean, and the 95% confidence interval. And so what you might be asked to do on the exam with this is you could be asked to calculate the mean for a given data set, and then you could be asked to, say, then take the standard deviation of that data set and potentially put that on a graph somehow. But a lot of times if you're asked to do anything with standard deviation, the chances are pretty excellent that the standard deviation will be given to you, because it does take a long time to calculate, especially if you do it by hand. And so just understand that you may be asked to calculate the mean of a data set that's given to you. And to answer Nate's question about the estimate of the number question on the exam about the labs, no, I can't tell you exactly how many questions will be about the labs because, for example, in the free response section, I wouldn't even say 25%. Really, it's not quite like that, because the distribution of questions is even among the 4 big ideas, but the science practices which labs are based on are kind of infused throughout. And so it's really hard for anybody to say, oh, well, there are this many questions about the labs. And so focus more on knowing, how to do science well, rather than, oh, this lab is going to be on the test. And so, don't focus so much on, oh, there's only this many questions about the labs. Focus on knowing how to do good science, and anything that comes up about a lab, you'll be able to answer. And I will address the 95% confidence interval thing. And no, it's not the same as the chi-square test. So let's talk about standard deviation, because standard deviation is going to kind of lead us into standard error of the mean and the 95% confidence interval. So remember that the standard deviation is based on what we call the normal distribution, or what we also like to refer to as the bell-shaped curve. If you've taken AP Stats or even any kind of statistics class, then you know that the standard deviation shows you how far away from the mean your data points are. In other words, it's a measure of dispersion. And so the standard deviation, this diagram that you see here, notice that it shows you that about 68% of all data samples fall within 1 standard deviation of the mean, and that the further out you go, the fewer data samples you're going to have under that curve. That you also need to understand this, is that you don't have to know how to calculate the standard deviation. You only need to interpret its significance. And so, calculating standard deviation, a lot of times, I know with my students, I have them do it in Excel, which obviously you're not going to have access to during the exam. And calculating it on the 4-function calculator that you're allowed to use takes a really long time. And so understanding the significance of standard deviation is the more important skill here, not so much that you have to calculate it. And so understand that standard deviation just means this is how far away my data point is dispersed from the mean. Now, in terms of, what does that really mean? If you have a large standard deviation, that means that, if you have a large standard deviation, I think I was going to say, it looks like my stuff got out of order. Okay, oh well. If you have a large standard deviation, then that means that your data points are very far dispersed away from the mean. And it means that there's a lot of variability in your sample. But if your standard deviation is really small, then that means that your data points are very tightly clustered together, and that there's not a lot of variance in your data. In other words, the data set is pretty uniform. And so, we can tell a lot about a group of data that we've gathered based on the standard deviation. But another way that we can do that is by looking at what we call the standard error of the mean. And the standard error of the mean is used to determine the error that exists in taking data from a sample of a population, because we can't ever really sample an entire population. Like, for example, if we were going to measure, in the the diffusion osmosis lab, for example, if we were going to measure the osmolarity of potatoes, we're not ever going to be able to measure the osmolarity of every single potato on this earth. We can only do a sample. And so inherent in that sampling error is the fact that we can't sample all however many billion trillion potatoes there are on the earth. We're only taking a sample of, say 24 potato cores. And so there's error inherent in that measurement. And so the standard error is a measure of precision. In other words, how good are our data? How reliable are our data? And so when we do standard error, we have to use the standard deviation as a part of that, which is why I'm telling you, if you had to calculate standard error on the exam, say, for example, in a free response question, they would give you the standard deviation. You wouldn't have to calculate that because it does take a long time. But you would have to calculate potentially standard error. And so the formula that's on your formula chart, so like I tell my students, please, for the love of all that is holy, don't memorize the formulas because they're on that formula chart. The standard error formula is the standard deviation divided by the square root of the number of samples in the data set. And so, pay close attention to any data table that they give you and counting the number of samples that are there. And like I said, they'll give you the standard deviation. And so that's what that's for. Now, the reason why the standard error is not a percentage. It is just an absolute value number, but it can also be, it's well, not absolute value, sorry. It can be positive or negative, because when we talk about plotting standard error on a bar graph, for example, you've probably seen graphs that have error bars. And so, when we put error bars on a graph, right, you place them above and below the center of the bar, right? And the reason we do this is we're comparing two groups. We're comparing the means of two group measurements, right? So like, for example, in this picture that you see here, they're comparing shady and sunny ivy leaf widths, right? And so, you see in the graph, the shady leaves versus the sunny leaves, and then on the left-hand side on the y-axis, you see the mean width of the leaf in centimeters. So automatically we're comparing the mean width of each of the leaves to one another. Now notice on the graph it tells you plus or minus 1 standard error. On an exam, they would tell you underneath, number one, if they gave you a graph, it would say plus or minus however many standard errors. If it was 1 standard error, they would tell you that. If it was 2 standard errors, they might also launch into something called the 95% confidence interval. And I'll talk about that here in just a second. But the error bars above and below the graph represent the standard error of the mean. In other words, we don't know where the true mean of our population is, but we know that it's between these bars. And the reason we don't know the true mean of our population is because we only took a sample. And so when they plot the standard error bars above and below that, mean, that's what they're getting at. You could be asked to plot that. You could also be asked to plot the 95% confidence interval. The 95% confidence interval is just 2 times the standard error. Now, what does that actually mean? Well, the 95% confidence interval, is this idea that because we can't know what the true mean of our population is, we can be 95% sure that it is between, the upper bound and the lower bound, as bounded by the error bars. And so, it just depends on, what you're being asked for. And to address the question about the error bar equal to 1, I would say no, that the error bar here is not equal to 1. And because if you read and I don't know if you can see it very well on your screen, but if you download the presentation later, obviously it's going to be a lot bigger. The standard error here is actually kind of small, because if you look at the increments on the graph on the left side, they're pretty big. They're in increments of 1. And so the standard error here is much smaller than that. And, I guess the disadvantage of seeing this graph is that you don't see, what the calculated standard error was. If I had to estimate, I would say that the standard error was probably like 0.25, just because it's pretty small on the graph. And so, the mean here, okay, to answer Anna's question, that a mean marked 6 could be 5 or 7. Yes, if the standard error were 1. And so here the standard error is not 1. The standard error here, like I said, I'm estimating that to be 0.25, which means my error bars go 0.25 above and 0.25 below whatever the value of the mean is here, which looks to be about 7.3 or 7.4. And so, that's how you would interpret that. And then again, if this were a graph with the 95% confidence interval, remember that what that tells us is that we're 95% sure that our mean falls somewhere within that range, but we don't know. And it's because we can't know since we didn't sample the entire population. And so you could see both kinds of graphs in the exam. You could be asked to produce both kinds of graphs in the exam. It just really depends on what you're asked. You could be asked to produce a graph like this, like if your teachers have given you access to the free response questions that are released to everybody publicly on AP Central, the free response question number 1 about trichomes and fast plants, that students were asked on that question to produce a graph that had, error bars on it. Most often, though, you're going to see graphs with error bars within questions, not only on the free response, but in the multiple choice as well. And so you need to be able to understand how to read them and understand what the graph is telling you by the presence of those error bars. Now, one of the most common questions I got asked was how to read them, and so when we talk about the error bars overlapping or not overlapping, this is the thing that's like super duper important. So when you have a graph and it's got error bars that don't overlap, right? So if you look here on this graph right here where it says enzyme activity and then you have control and treated, notice it tells you that the mean is here and it's plus or minus the standard error. Now they don't tell you what the standard error is. They're just telling you that what it says n equals 5, that was just their number of samples. So what they're showing you is a set of error bars that don't overlap. In other words, see that red line that I've got right there? I drew that red line, that horizontal red line, to show you that the error bar for the control doesn't overlap the error bar of the treated condition. And what this means is that there is a statistically significant difference between the means of the two groups. So what does this really mean? You need to do more statistical testing. That whatever statistical test you conducted, standard error, standard deviation, whatever, isn't enough. And so you need to conduct more statistical testing to get a conclusive result. It might also mean that you have to go back and do more experimentation to gather more data. Notice their sample size here was pretty small. It was 5. And so you do need to understand the significance of the error bars overlapping or not overlapping. So when they do overlap, as you can see in the second picture right here where it says the cell size of the cheek cells and the onion cells, notice if I were to take these two bars and physically lay them over each other, the error bars would overlap. And so what does that mean? That means that there's not a statistically significant difference between the means of the two groups. What does this really mean? It means you need to run more statistical tests. Because you may or may not find that a significant difference exists. It just means that you might need to run more testing, more statistical testing in order to determine if there truly is significance or not. And so please make sure that you understand what the significance of overlapping error bars is, what the significance of error bars that don't overlap is, because it is very common to be asked questions about, what is this data set telling me? And you would have to know based on, whether or not those bars overlapped. The other thing that you could be asked too, and I've seen this before in free response questions, it's like, okay, what is the significance of these results? And you would have to be able to talk about, how do the data display, how do the data show you that the results are not significant? Something to that effect. You would need to be able to understand how to interpret a graph that looks like this in order to determine whether or not the results were significant. And to answer Rebecca's question about the link to the presentation, I am going to put it in a Google Drive folder, and I'll link it to the description of this stream. So don't worry, you will have access to this, because I may actually go back and add a couple more notes to this presentation after I get done here today. So you will have access to it. All right, so making graphs making graphs, because this was something that a lot of students ask questions about in terms of like, how do I make graphs for the free response portion of the exam? And this is something that, I always tell my students, if you get a free response question and it has a graph, the graph is usually, worth about a third of the points of the question. You don't want to miss any of these points because they're very easy points to get as long as you know how to, produce a graph properly. And so I want to talk to you about that because these are some of the most easy points for students to pick up, but it's also, I think, important for you to know not only how to make the graph, but how to interpret it. And so on a free response question, you could be asked to produce a graph from a data set as a part of the question. And so constructed, when you make a graph properly, it has to have several things. And so, first of all, label your axes. They must have units. It is not enough to just label an axis with whatever the independent variable is and what the dependent variable is. So for example, if we were graphing, reaction rate of an enzyme reaction, on the y-axis you would have reaction rate. Well, don't just write reaction rate. Write the reaction rate as a function of whatever units over time were being, calculated. So please make sure that you get those units in there because students lose points on graphs for not putting units. I always tell my kids, no naked numbers. So don't put numbers on a graph without a unit. Make sure that you use SI units. It means metric units. So we're not working in the standard measurement system here. We're always operating in metric. So please make sure that you pay close attention to that. And so incrementation of your axis is also pretty important. And more importantly, that your axes are oriented correctly, that your dependent variable is on the y, that your independent variable is on the x. If you flip your axes, a lot of times that doesn't count. So please make sure that your axes are not incremented, that they're oriented correctly. Make sure that your points are plotted correctly. You'd be surprised at how students forget to do that sometimes. And that points should be connected if it's a scatter plot. If it's a bar graph that you're being asked to make, please make sure that you make a bar graph. In fact, there was a free response question, I believe it was last year, the very first one on the exam, about plotting the allele frequency of the LAP94 allele. And the question specifically said, create a bar graph. And students still made line graphs. So please make sure, and even some of my students did that, which was a little frustrating. But please make sure that if the question tells you to produce a specific type of graph, that you produce that specific type of graph. You don't want to lose points because you didn't follow directions. And so the last bullet point there that says the graph is the correct type for the data presented. If a graph is asking you to plot, say, for example, reaction rate, a bar graph is not the appropriate graph, especially if you have to calculate a change a change in reaction rate. You can't do that from a bar graph. And so, please make sure that you're plotting, that you're creating the graph that's correct for the data that's being represented. And to answer the question about making titles on the graph, a title is not necessary. If you go back, excuse me, my nose itches, it's allergy season here in Texas. If you go back and look at past questions and their grading schemes, the rubrics, nowhere does it say that students receive points for giving the graph an appropriate title. They do get points, though, for proper orientation of axes, proper labeling of axes with units, proper plotting of points. If a rate has to be calculated, that the rate is calculated correctly, that sort of thing. And so, you don't have to, you don't have to make sure that you have a title, okay? I know that that's something that your teachers have probably always imparted to you, but in terms of the free response question, we're more concerned with, are you communicating the results of the experiment correctly? Are you able to then interpret the results of the experiment correctly based on the piece of communication that you've generated? So hopefully that helps. Hopefully that helps you understand, producing graphs a little bit better. All right. Let's go ahead and move into experimental design because this is something else I got asked a lot about. Occasionally, you're going to be asked several things. You could be asked to design an experiment. You could be asked to evaluate an experimental design somebody else designed. You could be asked to propose a method for testing something. You could also be asked to pose a question that can be tested. And you could also be asked to state a hypothesis that can be tested. Um, all of these require, experimental design skills. And so a couple of the things that I want to go over with you, is just key elements of good experimental design. And so the first thing is a testable question, right? You want to make sure that the question you're asking is something that can be tested using scientific methods. It's not a rhetorical question. It's not, something that is just a what if this could happen. No, you have to be able to test the question using a scientific method. And those scientific methods include, having dependent and independent variables. The first one I want to talk about is the independent variable, even though I've listed it second. And the reason why I want to discuss the independent variable first is because this is the one that's manipulated by the experimenter to affect an outcome. And you're only going to have one of these at a time being tested. Now, does this mean you can't have multiple ones? No, you just can't have multiple ones at the same time. And the reason why is because if you have multiple independent variables being tested at once, then you don't know which one is affecting your outcome. In other words, which one is causing the outcome. And so you need to make sure that you're only testing one independent variable at a time. The dependent variable, though, is the variable that is measured. Okay, so this is what we're counting. This is what we're measuring. So for example, for the enzymes lab that probably every single person does, the independent variables are things like changing the temperature, changing the pH, changing the substrate concentration. Whereas the dependent variable is going to be reaction rate over time. And so I can change my independent variables, just not all at the same time. Like I don't want to do a temperature and pH experiment at the same time, because if I do that, then I'm not going to know which one of those independent variables is changing, it's causing my outcome. And so that leads me to the next thing, which is my conditions held constant. You always have to have conditions held constant, and you want to hold as many things constant as possible. And the reason why this is because it reduces what I like to call confounding factors that prevent you from collecting high quality data. In other words, if you hold all of these things constant, this is what's going to make sure that the independent variable is the only thing having an effect on your outcome. And so, you want to make sure that you're holding as many things constant as possible. So for example, with the enzymes lab, you would want to make sure that you had the reaction taking place in the same size vessel, that you were using the same volume of substrate, that you were using the same volume of enzyme, that you were using the same temperature if temperature wasn't your independent variable. Excuse me. Sorry. You would want to hold as many things constant as possible. The control group is not the same as a constant. I see that a lot. As an AP reader, I see that a lot when I've graded questions that had to do with experimental design. Students confuse control and constant all the time. And so I want to make sure that you understand the difference between the two. The control group is the standard for comparison. In other words, this is my group that doesn't receive my independent variable. Okay? So, for example, if we were doing an enzymes lab, and the only reason I use that one is because it's a convenient example. The enzymes lab, for example, that my students and I did, we used yeast spheres, and we reacted the yeast spheres with hydrogen peroxide at different temperatures and different pHs, but we also used different strains of yeast to see if that would change the reaction rate of the yeast catalase that was in the yeast with the peroxide. The control group in that instance, for all of the experiments that kids did, was a yeast sphere in water, in distilled water, because there's going to be no reaction there at all. And so that's going to be the standard that we compare everything else to. And so, make sure you understand that the control group is not the same as a constant. They are definitely not interchangeable. So please make sure that you keep those two things straight. Another key piece of experimental design is repetition. You have to have multiple trials. You can't just run an experiment once and say, okay, I can have conclusive data from just this one iteration of the experiment. No, 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 no. You have to have multiple trials. In the past, when they've asked questions about experimental design on the free response question, repetition is one of the things that we give points for, that somehow the student indicates that the experiment was collected, that data was collected more than once. And so, make sure that you include multiple trials. If it's an experiment about evolution, that something takes place over multiple generations. Just the importance of multiple trials cannot be stressed enough. And so to answer the question that Anna asked about what would a control group be in an experiment with different temperatures as the independent variable, your independent variable would be the various temperatures, but your control would be room temperature. So the control group would be whatever your reaction is going to be at room temperature is what my students and I always do. And so hopefully that answers that question for you. And so make sure that you understand these elements of experimental design, because again, you could be asked to evaluate somebody's experimental design based on these things. You could be asked to propose an experiment based on these things. So please make sure that you understand, all of these, items that go into experimental design. Okay, and so evaluating an experiment, look for the elements of good experimental design, the ones that we just discussed. Is there only one independent variable being tested at a time? Is there a control group for comparison? Is the experimenter conducting repeated trials? And one bullet that I didn't include on here, but I'll go back and put it on there. Did the experimenter hold conditions constant so that the only thing affecting the outcome is the independent variable that they tested? And so, please make sure that you really, really understand those key pieces, those key elements of good experimental design, because you could be asked to evaluate an experiment. I've seen them ask that a lot, especially in multiple choice questions. And so, not so much on the free response, but definitely in the multiple choice questions. So as long as you have, those key elements of design understood, you should be okay to evaluate experiments, to generate experiments of your own, to propose questions that are testable, that sort of thing. So let's talk about some labs, because there was a cluster of labs that students asked about over and over and over again. And so, I want to make sure that I discuss those. And most of them were under Big Idea 1. And Big Idea 1, remember, is evolution. And so the labs that you might have done as a part of this big idea that your teacher taught you, you might have done the artificial selection lab with the fast plants and the trichomes. You all probably did a Hardy-Weinberg lab of some sort, whether it was the computer modeling or some other kind of modeling, and the blast lab, or some sort of lab that taught you how to make cladograms. And so let's talk about the artificial selection lab. And the reason I talk about the artificial selection lab is partly because of the concept that it's designed to investigate. So notice that the question investigated here is, can extreme selection change expression of a quantitative trait in a population in one generation? In other words, can selection change the expression of a trait that I can count in a population in just one generation? And so the concept here is that you need to understand that, natural selection acts on phenotype. Right? Natural selection acts on phenotype, but ultimately that selection leads to changes in genotype. Natural selection is also a major mechanism of evolution. And so the setup for this lab is pretty easy. You grow a set of fast plants for several generations, and you select each generation for the number of trichomes. And then you breed those guys. You find the ones with the highest number of trichomes, selectively breed them, count the trichomes of the plants over each generation, and collect data. Okay? And so the expected results that you should see is that directional selection should take place. In other words, now we're shifting from, one extreme, maybe no trichomes, to the other where we have a lot of trichomes, or even kind of pushing it to the middle and then move over to the end. And so that's going to change over time as we increase the number of generations. I know that I have some colleagues that do this lab all year long, and so they've generated, 4 or 5 generations, depending on when they started school. An exam question about this, free response question number 1 on the 2014 exam. If you look at AP Central, it's there for you to look at. It's a released exam question. And so that first question was about trichomes. It also included making graphs that included error bars. So the things that we've already talked about were kind of rolled into this lab question. So that's what that lab is all about. Now, the one I got the most questions about was the Hardy-Weinberg modeling, because students seem to have some confusion about what Hardy-Weinberg was all about. And so the question that's investigated in any Hardy-Weinberg lab that we do, regardless of how it's done, is how are mathematical models used to investigate the relationship between allele frequencies and populations of organism and evolutionary change? Because one of the things that's super important that you need to know about Hardy-Weinberg, it is a mathematical model. It's a mathematical model that's used to determine when evolution happens in a population, because the conditions of the Hardy-Weinberg equilibrium are for a population that doesn't evolve. And so we use it to compare real populations to, so that we can see, oh, hey, this population's evolving because the allele frequencies are changing. And so the concepts in this lab are that, natural selection acts on phenotype, like I said before, which leads to changes in genotype. Natural selection is a major mechanism of evolution. Evolutionary change is driven by random processes like genetic drift. And populations of organisms continue to evolve. Okay. And so for this lab, the way some people do this lab, and I don't do it this way, so I've done it this way before, but I don't do it this way now with my students. They set up a spreadsheet, okay, usually Excel, usually you can also use Google Sheets for it. But it sets up a mathematical model in the spreadsheet that allows you to see changes in allele frequencies over time. Okay, and so students are given certain scenarios like natural selection, genetic drift, gene flow, that sort of thing. And the spreadsheet has to be altered to reflect those changes in allele frequencies. And so the biggest thing that you need to be familiar with here is this equation. P squared plus 2pq plus q squared is equal to 1. Where P plus Q is equal to 1, and the reason why P and Q are equal to 1 is this. Remember that P is the frequency of the dominant allele, Q is the frequency of the recessive allele. And the reason why P and Q equal 1 is because the probability of inheriting either one of those alleles is 1/2. And so 1/2 plus 1/2 equals 1. Now, when you look at that P squared plus 2pq plus q squared equation, P squared is the frequency of the homozygous dominant phenotype. 2pq represents the frequency of the heterozygous phenotype, or not phenotype, excuse me, genotype. And back this train up. P squared is the homozygous dominant genotype, my bad, because P squared plus 2pq is the dominant phenotype. Q squared represents the homozygous recessive genotype. Now, students also sometimes don't see the connection about the equation, like where it comes from. So that's why I included that graphic there of the Punnett square. Notice if I put my P and Q into a Punnett square representing the fact that there's a 50% chance of inheriting either one of those alleles because they're, one's dominant, one's recessive. Look at the beautiful Punnett square that results. You get P times P gives me P squared, and I get 2pq, and then I get Q times Q gives me Q squared. And so make that connection between the model of the Punnett square and the equation itself, right? And the fact that you have a 50% probability of inheriting either a dominant or a recessive allele. And that's a connection that sometimes students don't make. And it's when this equation as it's written is, when those allele frequencies don't change, our population is not evolving. And so the population evolves, though, when one of the following things happens. So, for example, to keep a population in a Hardy-Weinberg equilibrium, in other words, to keep it from evolving, okay, there can't be any selection. My population has to be really, really big. Mating's got to be completely random. There can't be any mutations, and there can't be any gene flow. So what does that really mean? I can't have any natural selection. Why? Because it favors one trait over the other. Mating has to be completely random. Why? Because if we're choosing our mates for a particular reason, we're doing selection. In fact, we're doing sexual selection when we do that. Mutations, We can't have any mutations if our population is going to be in equilibrium, because a mutation might confer a favorable trait upon, a group of organisms. Also, it could also confer an unfavorable trait, and that would lead to selection. And so, no mutations can occur. My population has to be sufficiently large to avoid sampling error. And then you can't have any gene flow either, because remember, folks, when genes flow, that means you're either taking your DNA out of the gene pool or you're putting it back in. Because remember, like I always tell my students, when organisms migrate or immigrate or emigrate, they either bring their DNA with them or they take it away. And so if I'm taking my genes out of the gene pool, then I'm changing the allele frequencies of that gene pool. And so if all of these things happen, allele frequencies are not going to change and evolution is not going to occur. And so if these things do happen, like if mating isn't random, then I'm going to have evolution of the population. If mutations happen, I'm going to have evolution. If I have gene flow, I'm going to have evolution. And so that mathematical model, this is just a way to quantify changes in a population's allele frequencies over time. And so hopefully that gives you kind of a good foundation for how to understand that. Now, the question is, a lot of kids ask, do we have to know how to calculate Hardy-Weinberg? I'm going to say yes, you do, because I've seen it show up on exams in the past, but I think it's more important for you to interpret the results of a Hardy-Weinberg experiment or a Hardy-Weinberg situation. And so please just make sure you understand that when allele frequencies change over time, evolution is occurring. If my allele frequencies change, then, the thing is, my population is no longer in Hardy-Weinberg equilibrium. And so to know the difference between Q and Q squared in a word problem, Q squared is remember, it's going to be the recessive genotype. So if they say, this many organisms out of this many have the recessive genotype, then you would set Q squared equal to the number of organisms with that genotype over the number of total organisms. And so, for Q squared, it's going to say it's the recessive genotype and phenotype for those individuals. And so, it'll say Q, it'll say allele frequency. That's right. Exactly. Very good. So let's talk about BLAST, because this was something else I got asked a lot about. Not so much the BLAST lab itself, but about cladograms. And so the BLAST lab, I know a lot of teachers do this. I'm going to be real honest. I don't. I do a different lab that teaches the same concept, but I don't use the BLAST to do it. And so the question investigated here is, how can bioinformatics be used as a tool to determine evolutionary relationships? And so your key concepts in this lab are going to be that natural selection acts on phenotype, just like it is for all of the evolution labs. That evolution is supported by scientific evidence from many disciplines, including math, because you do use a lot of math here. Phylogenetic trees and cladograms are models of evolutionary history that can be tested. And that DNA, and in some cases RNA, is a primary source of heritable information. And so the setup for the BLAST lab is basically this. You're given a set of DNA sequences, and you're given the BLAST database to determine the evolutionary relationships among a group of organisms. And so that database is actually a really, really powerful piece of software that collates all that information of the DNA sequences, finds commonalities between them, and then spits back information to you about percentage similarity, percentage difference, and all that kind of stuff. And so then you would use the data that's gathered from the BLAST website about the similarities between the genes and the protein sequences for this set of organisms to create the cladogram that shows evolutionary relationships. And so you use that information found in the BLAST database to compare those gene sequences from an unknown fossil to genes of existing organisms, so that you can place that fossil organism on a cladogram with organisms that are currently alive. And so, like I said, I don't do this lab as it's written. I do a very different version of this lab that uses a different database but shows the same idea, where we're looking at protein sequences to see what the commonalities are between organisms that we study a certain protein, for. For example, we look at the hemoglobin beta protein and we compare the amino acid sequences of that protein for several different organisms. And so when you create cladograms from data, there's a couple different things you need to be aware of. First of all, you could be given a data table that shows you the number of amino acid sequence differences. You could also be given a data table that shows the number of DNA differences. It just depends. It depends on what they give you. Regardless of what they give you, you have to be able to put that information into a cladogram. You may be asked to draw one. You could also be asked to place organisms on a template. If I remember correctly, I believe it was 2014, question number 2, asked you to do that based on differences in protein sequences of milk proteins for a group of mammals. And so, please make sure you understand that when you place organisms on a cladogram, like for example, if you had to draw one, so the cladogram you see here shows divergence away from a lamprey based on hemoglobin, right? And so notice the vertical axis there on the left is time, and if we're going across the bottom, that shows, the number of differences in amino acid sequence away from the human. And so the lamprey is going to be the furthest away from the human because it shows the most number of differences. And then the frog, and then the bird, and then the dog, and then the macaque, and then the human. And then notice the human and the macaque share kind of the same branch because that branch point indicates common ancestry between those two. And so anywhere you see, a branching point, so like right here, let me see if I can draw a little circle to show you. Like this would be a branch point, and then this would be a branch point, and then here would be a branch point, so on and so forth. And so each one of those would represent a place where divergence had occurred, speciation had occurred. And so be able to, number one, interpret a cladogram, but also be able to produce one if you are asked. And so to find common ancestry on a phylogenetic tree, you would actually have to look at the same branch points. It's just that the phylogenetic tree is organized slightly differently. And so make sure you understand that, that's where you would find the common ancestry, is it would be at the branching point. And so to answer the question about background information, if it's a lab. Okay. So the question asks if it gives, the question's being asked says, would it have background information if it's about a lab? All of the questions are going to give you background information, right? Like there's a lot of reading. And so that reading is about, it's providing you context. And so if the free response question asks you for an example about a lab, here's the thing. It's going to ask you about a lab you may not have ever done. It's going to ask you about evaluating a lab that somebody else did. It could also ask you to maybe improve their experimental design somehow. And how you would know to do that is to, know the principles of experimental design. Okay? Know those principles of experimental design. As long as you know those principles of experimental design, you could design any lab. It doesn't matter. And so, it doesn't matter what you're presented with. And so it just really depends on, what kind of context they're giving you and all that. So don't freak out about, oh my gosh, I've never done this lab before. It's okay as long as you understand the principles of experimental design. So just kind of getting back on track with cladograms and whatnot. Like I said, you could be asked to evaluate one. You could be asked to produce one. So be familiar with, how to read one, how to produce one based off of data that they could give you, that sort of stuff. Let's see. So these are just for this particular example. This is just showing you amino acid differences. This isn't actually showing you gene differences, and so because it's not giving you the DNA sequence, and so it's not showing you how many A's versus G's or anything like that. It's actually just showing you the amino acid sequence differences, and so most often you would be given amino acid differences rather than, differences in DNA, hopefully. All right, at least that's how I've always seen it. It was differences in amino acid sequence, not differences in DNA. All right, so let's talk about labs and big idea too. The most commonly asked about lab was the diffusion osmosis lab. Surprisingly, nobody asked about photosynthesis or respiration. And so I do want to focus on the diffusion osmosis lab, because there were lots of questions about water potential. And so I want to make sure that you understand the concept of water potential and how to calculate it and all that kind of stuff. So the question that's investigated in this lab is, what causes plants to wilt if they're not watered? And so the concepts here are that, growth, reproduction, and homeostasis require that cells have to have internal environments that are different from their external ones. In other words, we got to have a membrane. We got to have a membrane that separates our outside from our inside. And then that membrane is going to regulate, what goes back and forth across the membrane. And so, cell membranes are selectively permeable. Diffusion osmosis are forms of passive transport that move molecules across membranes. And that water potential is the potential for water to do work. And when I say that water is doing work, it's doing the work of dissolving solutes. And so water potential is really a measure of the free energy of water. Okay? And just really quick, I'm looking at the questions. Do you draw the amino acids differences on the X's? No, you don't. You don't have to. In fact, it's preferable probably if you don't. It would be a waste of your time to do that. So let's talk about diffusion osmosis, and then if I have time, I will kind of give you a quick and dirty review of the photosynthesis lab, but if not, I'll put it in the presentation for sure that you can download later. So the setup for the diffusion osmosis lab, the first part of it is that you have artificial cell models that explore the relationship between surface area volume and the rate of diffusion. So you might have had little cubes that had phenolphthalein in them, and you poured sodium hydroxide over them and they turned pink. You might have also had ones that turned blue. It really just depends on what chemical your teacher used to make the cubes. I know my students and I do this lab using probeware with cubes that have salt in them, and we measure the conductivity of the solution that the cubes sit in. And it shows the same exact thing. But the second part of the lab is you're exploring water potential. Knowing molarity of all solutions, no, you shouldn't have to know how to do grams to moles and all that. This isn't AP chemistry. A lot of times the molarity of the solution is given to you in the problem. And so exploration of water potential, basically you cut plant tissue cores. You might have cut cores, you might have cut cubes. It just depends on what your teacher had available to you. You soak them in sucrose solutions of varying molarity to determine the sucrose concentration of the plant tissue. And so what you've got here in the picture is the fun little cork borer, piece of potato cores. Again, this just depends on what plant tissues your teachers had you use. I know we used potatoes and carrots and beets and squash and all kinds of stuff, sweet potatoes. We used a lot of different things. And so the end result is pretty much the same. Like you're looking at a change in mass of those plant tissue cores with increasing molarity. And so let's talk about the expected results. So for the first part of that lab with the cubes, you need to notice that if the cell gets larger, the surface area to volume ratio decreases. And so this little graph over here on the right kind of shows that relationship. That as the cube gets larger, that surface area to volume ratio decreases, which means that the cell is going to have a harder time performing the functions of life, basically, because I always explain it this way, the further away my nucleus is from the cell membrane, the harder it is for materials to get in and out of that cell, for materials to get in and out to the nucleus, for the cell to be able to perform all of the functions that it has to do. So when my cell is really small, notice my surface area to volume ratio is really, really large. And so you also need to understand that cells have adaptations for increasing their surface area to volume ratio. They can be large, but if they're going to be large, they're going to be flat. Or if they're going to be large, they're going to have a lot of invaginations and stuff like that. In other words, they're going to have a lot of folds that that increase the surface area, kind of like a terry cloth towel. That's what I always explain to my students, that a terry cloth towel has loops on it to help you absorb all the water from your body when you take a shower, you get out and you dry yourself. And so, sorry, my nose is really itchy. And so that first part of the lab is all about surface area to volume ratio and how cells can exchange materials with their environment based on that surface area to volume ratio being very, very large is what you would ideally want it to be. It also kind of goes back to the whole why are cells small. Cells can be small, but they can't be too small. You don't want them to be so small that they can't fit all the machinery inside, but they need to be small enough so that they can conduct the business of transporting materials back and forth across the membrane. Now, water potential. This is the thing I got asked about the most. First of all, water potential is going to be the measure of how much free energy water has available to do work. And when I say the work, I mean, work of water, I mean the work of dissolving solutes. And so, water will form like these little shells of hydration around solutes. And once a water molecule is occupied, in that shell of hydration, because it forms like loose hydrogen bonds with that solute, it can't bond to anything else. It's occupied. And so when I add solute to a solution, I drive my water potential down because now I've got water molecules occupied that can't do the work of dissolving solute. And so, water potential is this concept basically that says, okay, when I have a lot of solute in my solution, my water potential is really, really low. When I don't have a lot of solute in my water solution, my water potential is really, really high. And the reason why this whole idea of water potential even matters is this. Plants get water because of changes in water potential, or due to differences in water potential. The root of a plant, by its nature, is hypertonic. In other words, it has a high solute concentration and a low water concentration. And if you think about it, it makes sense because roots are designed as storage organs. They store carbohydrate. And so there's going to be a lot of sugars and a lot of starches in that root. Well, that means that the water potential of that root is going to be incredibly low in comparison to the soil water around the root. So water will move from the soil into the plant. And then when water moves from the soil into the root, pressure builds in the root and it pushes it up the xylem tissues of the plant toward the leaves. And this kind of goes along with the transpiration lab because transpiration also occurs because of differences in water potential. When water moves up those xylem tissues and it goes out to the leaves where it exits the stomata, then basically what happens is that, water is moving from an area of high water potential in the leaves to the atmosphere, to the air where the water potential is very, very low. And so, kind of think about water potential, not just in terms of, osmosis, but also in terms of transpiration. And so for these, potato cores, beet cores, whatever you used, you need to understand that the plant tissue is going to gain mass in a hypotonic solution. In other words, a solution whose water potential is really high. And plant tissue is going to lose mass in a hypertonic solution. And so notice that the graph shows you the change in the mass of the cores, and usually it's done in percent, it's a percent change, and it increases, the plant tissue mass changes as we approach, okay, let me back up. The percent difference is positive until you cross the x-axis. And the reason why is because those first few solutions are what we would consider to be hypotonic. In other words, they have a high water potential. So water is going to move from those solutions into the plant tissue. Once we get to the x-axis, though, that's where we hit what we call an isotonic point. In other words, this is where the water concentration of my plant is the same as the water concentration of my solution. And so that point on the graph is pretty important because that helps us determine the concentration of solute in our plant tissue. And so anything below that, though, is showing a decrease in the percent change of mass because our plant tissue is losing mass. Now, to answer the question about water potential ever above zero, water potential can be above zero if pressure potential is positive, if there's pressure potential. Because the formula that we use most often is this one, right? So if we're calculating water potential from the experimental results that we got, here's your formula. Psi equals negative ICRT. And so, the water potential formula in general, though, is pressure potential plus solute potential. Solute potential is that ICRT, that negative ICRT. But in most instances, pressure potential is zero because they always tell you, oh, the container is open. Oh, it's at atmospheric pressure, which means the pressure potential is equal to zero. So pressure potential can be, a value if it's given to you. And so that can cause your water potential to be over zero. Like, for example, if we had, a piston pushing down on a beaker or a syringe or something, that would give us a positive value for water potential. Potentially could give us a positive value for water potential. And yes, the variables are defined on the formula sheet. And will it ask for water potential with ionic solutes? Yes, it can. And so if it asks for the water potential, if it asks you to calculate water potential for something that has an ionic solute, then what you need to know is that you need to understand how many ions that solute breaks up into. So for example, if it's salt, then it would be, 2. I would be 2 because it breaks up into sodiums and chlorides. And so and that's correct. The ionization constant for sucrose, and really I like to say to my students, anything that ends in OSE is going to be 1. Sucrose, fructose, glucose, maltose, whatever ose, it's always going to be 1 because those things don't ionize because they're covalently binding compounds. So if the cell has a higher water potential than the solute, is the cell hypertonic? Yes. No, 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 sorry, no, because water potential, look at it this way. Water potential, the more solute you add, the more negative it becomes, okay? And so when my water potential is high, my water potential is more positive. It's closer to 0, okay? When my water potential is low, it's further away from 0, okay? And so if my cell has a high water potential, then my cell is hypotonic, not hypertonic. And so a high water potential is closer to 0. A low water potential is further away from 0, right? And so if we have to calculate water potential, like I said, here's your ionization constant, which is going to be 1 for things that end in OSE. C is going to be the molar concentration of the solution. Now, if you were asked to get it off of a graph, you look for the place where the line crosses 0. And so, let's see, the circle, yeah, there we go. You look for that spot where the line crosses 0. And let me kind of make that transparent so you can kind of see. There we go. You look where the line crosses 0 because that's going to be where your concentration of water is the same inside the solution and inside the zucchini or inside the potato or the whatever, and that's going to represent the concentration of solutes inside your plant. R is a constant. Remember, it's the pressure constants. It's 0.0831 liter bars per mole K, and then temperature has to be in degrees K. And so you're often given the temperature in degrees C, so just remember to add that to 273 to get that value. And then you would have to calculate that out. Do not forget your signs. I noticed that when I gave my students their mock exam this week, there was a water potential question on it, and they forgot their signs. They did all their math right, but they forgot the sign. They forgot the negative sign. So please don't forget that negative sign, because remember, water potential is most often going to be negative because you are decreasing the ability of water to do work by adding solute to a solution. And so it's going to be a negative value. Okay, it's going to be a negative value. All right, one of the other labs, potentially, yes, you might have to do calculations on the free response part. It just depends on the question. All right, one of the labs that kids asked about in terms of Big Idea 3 was the restriction enzyme analysis, the electrophoresis lab that pretty much all of us do. Surprisingly, nobody asked about meiosis and nobody asked about transformation. And so I want to talk about restriction enzymes. So, the question that's investigated in this lab is how can we use genetic information to identify and profile individuals? Your concepts here are that populations continue to evolve, that DNA is the primary source of hereditary information, that the chromosomal basis of inheritance provides an understanding of how genes are passed from parent to offspring, and that restriction enzymes recognize certain sequences of DNA and will cut the DNA at those sites, creating fragments of varying size. The other thing you have to remember, too, is that those restriction sites are scattered at random throughout somebody's genome. And so no two individuals are necessarily going to have the same exact restriction fragments that are generated from a restriction enzyme digest. This is how we're able to use this particular technology to identify individuals. And so the setup for this kind of looks like this. You make a gel. You obtain DNA samples from whatever it is you're going to study. Those DNA samples have then had restriction enzymes added to them so that they get digested and chewed up into the different size fragments. You put those samples into the gel, and then you separate those fragments using electrophoresis. Remember that you load the gel such that the DNA goes into the negative end of the electrophoresis chamber. And then once that chamber is filled up with buffer and the gel. You apply an electric current for a certain specified amount of time. It just depends. And then after that time, you stain the gel so that you can visualize those fragments, because DNA is not colorful. It's clear. And so you have to stain the fragments so that you can see them. You measure how far away from the well they traveled, and then you create a standard curve based on your standard ladder there on the far left, or excuse me, the far right. Normally it's on the far left. No, you're not going to be able to tell the orientation of the DNA 5' 3' in the gel. That's not what that's for. What it is for, though, is showing you that the restriction enzymes will digest the DNA into different size pieces, and that those pieces of DNA will settle at different points in the gel based on their size. Okay, but it's also a function of how concentrated the gel is and how much the the voltage of the electrophoresis chamber and all of this other stuff. But the important thing is bigger fragments are going to be closer to the well, smaller fragments are going to travel further away, and it's because they're smaller and can wind through the gel a little bit easier. And so what does this show us? Here's an example of a gel that was run. This particular gel shows samples of DNA from individuals who have normal hemoglobin, which is going to be this. And the normal hemoglobin is going to be cut by a restriction enzyme. Why? Because it has restriction sites that are recognized by the restriction enzyme that was used in this instance. Okay. This also shows somebody who has sickle trait, so they have a normal allele. They have an allele for normal hemoglobin, but they also have an allele for sickle hemoglobin. And notice that the sickle hemoglobin sample doesn't get cut at all, and it's because there's a mutation in the DNA in the restriction site that the enzyme would recognize, and so it doesn't cut it at all. And so somebody who is homozygous for their normal hemoglobin would have 2 bands. Somebody who has sickle trait would have 3 because they have 1 allele for mutant DNA and 1 allele for normal DNA. And then somebody who has full-on sickle hemoglobin doesn't have a cut at all. They don't have a restriction site because there's been a mutation that interrupts the sequence that was cut. As far as knowing specific steps of the PCR, I think I would understand the principle behind it, that basically you're taking DNA and using an enzyme that's reserved for organisms that live at really high temperatures. You're replicating DNA ex vivo. In other words, you're doing it outside of a cell. But in terms of like knowing the, the separation, the annealing, the denaturing, the annealing, the extension, I don't think you need to know that. Just understand what that process is used for, that it's used to create lots of DNA in a really short amount of time. Because often PCR precedes this. PCR often precedes electrophoresis, because let's say, for example, we're pulling DNA from a crime scene. We might not have a whole lot of DNA to work with, and so we isolate the DNA from the samples we take at the crime scene and then amplify it using PCR so that we can then run a gel like this to identify individuals that may or may not have been present at the crime scene. All right, just super quick here. The labs you might have done with a big idea 4 are going to be energy dynamics. Any uncut DNA belongs to a mutant. Potentially. It really just depends on the situation that you're being presented with. But most of the time, mutant DNA won't be cut by a restriction enzyme. Just because the site might not be recognizable by the enzyme. It really just depends. Excuse me, my nose is super itchy. I'm sorry. It really just depends on what the enzyme is that's being used and what gene we're looking for and all that kind of stuff. All right. Big idea 4 is interactions. And so labs you might have done here are going to be the enzyme lab that I talked about earlier, animal behavior, transpiration, and energy dynamics. The one that I want to focus on, though, is animal behavior. And the reason why I want to focus on this one, because this is one that they've asked animal behavior lab questions before, but they've asked them in an experimental design context, because this is a lab that typically teachers will do with their students in order to introduce them to experimental design principles. And so that's one of the reasons why I wanted to talk about this one in particular. Lots of teachers do animal behavior labs. It just depends on what animal they use. Some teachers use fruit flies, some teachers use pill bugs, some teachers use jewel wasps, some teachers use mealworms. It just depends on what they use, but really the principle behind the lab is the same. What environmental factors are going to trigger a response in these organisms. And so the concepts here are going to be experimental design principles, interactions within populations influence patterns of species distribution, and that the timing and coordination of behavior is regulated by a lot of different mechanisms, and they're important in natural selection. And so the setup, like the lab that my students and I do, we use pill bugs. And so we basically have a choice chamber that has a wet side and a dry side. We start off that way. And then, we, let them, we have another setup where there's 2 wet sides or whatever. And we have multiple pill bugs in each side. You don't want to just have one, because if you just have one, your sample size is really too small. So you want to make sure that you have an ample sample size. And then we allow them to move back and forth for a certain specified period of time. And we record the number of pill bugs on each side of the chamber at certain time intervals. And so, students will also elect their own variables rather than just wet and dry. Sometimes they'll put food on one side and not food on the other side, or they'll put a texture on one side but not on the other side. They'll make one side light and one side dark, one side warm, one side cold. It really just depends on what they want to do. But the idea here is that you want to make sure that you're employing those experimental design principles. Also, one of the things that students will do with this particular experiment is they'll run a chi-square test to determine whether or not they found they could predict how well the pill bugs would distribute themselves to either side of the chamber. There was a question, I believe, on the 2013 exam. I want to say it was question number 2, that uses a chi-square test in an animal behavior lab and a choice chamber, actually, to determine whether or not the experimental design was good. And so if you'll go back and look at that old exam, it actually is an excellent, excellent question about not just this lab, but about experimental design principles in general. And it uses the chi-square. And for that particular question, if I remember correctly, the table was mostly set up for students, and that students just had to carry through the calculation. They had to know how to determine the expected values. And the expected values for that particular question were determined by, here's how many pill bugs, or here's how many beetles, or whatever, I can't remember what the bug was. Here's how many animals there are. They're going to be evenly distributed to all 3 chambers. If our null hypothesis says that there's going to be no difference between what I expect and what I observe, then they're going to be evenly distributed to all 3 chambers. And so for this type of lab with independent variable glucose, why do you say? Okay, so think about it like this. To answer that question about glucose preference and why do we use water instead of leaving it dry, how do we know that the bugs aren't attracted to the water? How do we know that the bugs are attracted to the sugar? We don't know. And so we have to wet both sides of the chamber, one side with a wet cotton ball that has just water, and one side that has the wet cotton ball with glucose. Because if you leave it dry, then we really don't know what's causing the outcome. We don't know if it's the dry, we don't know if it's the sugar, we don't know if it's the wet. And so these are from, I want to say they come from BioCoach, or no, LabBench. I'll put a link to it in my presentation. And here the independent variable is not time. The independent variable is what you're changing in the environment. And so, time is just something you're measuring. Like if you do this over 10 minutes, that's not your independent variable. Your independent variable is going to be the wet side versus the dry side, or the glucose cotton ball versus the wet cotton ball. It's what are you changing in the environment to affect the outcome. You're not changing time to influence their behavior. You're changing the environment to influence their behavior. And so you want to make sure then you, when you try to interpret what the independent variable is here, it's whatever you're doing to the environment to influence the animal's behavior. It's not time. So hopefully that kind of helps explain that. And so the last thing I want to just kind of impart to you before we go for the day, always look for the patterns in the data, because you're going to be presented data on both the multiple choice part and the free response. So always look for those data patterns. I was teaching my students the other day, with those long reading questions in the multiple choice, always look at the data first. Don't look at the paragraph, look at the data first. What are the patterns? What are the trends? What is it telling you? Then go look at the question, because the paragraph is usually providing context. And so look at the data first. And then if you see models, and models, remember, are pictures, they're diagrams, they're data sets, whatever. What biological process is this representing or attempting to represent is the question you need to ask yourself. What are they trying to show me here? And then what are the limitations of the model that they're showing me? Because remember, models aren't perfect. They're useful, but they're not perfect. And so make sure you understand that, models are just representations of things that happen in nature, and they're representations of phenomena. And so make sure you understand that. And finally, remember, you've worked really hard all year long. Give it your best shot on Monday. I was going to say, show us the biology that you know. It's one day out of your life. It's one test, and you guys are going to be great. You guys are going to be awesome. And I hope you have success on Monday. It's going to say good luck. Make sure you get some rest. I will put the link to this presentation in the description here in just a little bit. I do want to add a couple things that you guys made me think about while we were talking. But it's been a pleasure working with you all today and presenting for you all today. I apologize for my itchy nose. The pollen counts here are kind of crazy. So I'm sorry if I keep scratching my face. But hopefully you guys are going to be ready to go on Monday. Just don't stay up too late tomorrow night because you don't want to oversleep your exam. So I'm going to go ahead and sign off. I just want to say thanks to David Konufke for getting this all set up, and Bob Kuhn, and my fellow presenters, Stephen Traphagen, Brittany Frankowkak, Ryan Reardon. I know I'm leaving some people out. I don't know who. But thanks, guys. It's been great. This was awesome. I really enjoyed it. So, all right. I'm going to go ahead and stop the stream.